Okay, so now we're going to talk about the complement system because you should understand the basics before we go into the details of the acute inflammatory response. So a fair discussion of the complement system is necessary. So the complement system is a part of the innate immune system, and it is composed of certain proteins that are found within the blood in an inactive form. When they encounter a pathogen, they work to eliminate that pathogen. But first, they have to get activated. And there are three different ways by which complement activation occurs. Now, in this video, we're going to discuss the classic pathway. Okay? Now, let us suppose that this is a pathogen right here. Of course, a pathogen expresses certain antigens on the surface. And those antigens, they bind with certain antibodies. Okay. Now, when we talk in the classic pathway, when we talk about antibodies, there are either IgM or IgG antibodies. Okay. Now, each antibody has something that is called an Fc portion. Complement proteins are within the bloodstream. Before that, I should tell you that each complement protein is composed of two portions, an A portion and a B portion, with the exception of the C1 complement protein. It is only one portion, so it does not get divided. Now, so C1 attaches to that Fc portion of the antibody. So this is C1. Okay? Now, then it attracts C4. And of course, C4 is comprised of A and B portions. So actually it splits C4 into C4A, which goes away, and C4B, which stays attached to the C1. So this is C4B. Now, this attracts C2 in the same way, C2A and C2B, and then it will split into C2A, which goes away, C2B, which remains here. Okay? Now this is an important thing to mention. This sequence right here, the C4B, C2B, this is called C3 convertase. Okay? So of course, as the name implies, they split C3 into C3b and C3a, which goes away. Now this sequence is also an important sequence because this is called C5 convertase. Okay? Now, C5 convertase, of course, it splits C5 into C5a and C5b. Now, this is an important point to understand. C5b has the ability to bind with C6, C7, C8, and C9. Okay? And when they bind, they cap off out of this sequence. They form a circular arrangement of molecules. Okay? Such as this arrangement, for example. So that circular arrangement of molecules basically falls on the surface of the bacteria. Now, there is no enzymatic method here. There's no enzymatic activation. They just disrupt the plasma or the membrane integrity on the bacterial surface, and that disruption leads to a difference in osmolarity and unwanted things could go into the cell, wanted things or essential things could go out of the cell. So basically the cell dies. And this is called membrane attack complex. Okay? This is called a membrane attack complex. Now, as we mentioned, there is no lytic enzymatic activity here. They just disrupt the physical barriers. They just disrupt the physical barriers of the membrane. Now, if someone were to ask a question, would this membrane attack complex be more efficient on gram-negative bacteria or on gram-positive bacteria? Now, a difference between those two is that this attacks lipid membranes. Okay? Now, let me just tell you a little quick difference between gram-positive and gram-negative bacteria. So this is a gram, let's say that this is a gram-negative bacteria, and this is a gram-positive bacteria. Okay? Now, they both have this lipid coat, this lipid membrane. Okay? However, and they both have something that is called a peptidoglycan. And this peptidoglycan is a surface, as the name implies, proteins that surrounds gram-positive bacteria, but is very thin in gram-negative bacteria, because gram-negative bacteria has another lipid coat outside. Okay? So the MAC would basically be more effective on gram-negative bacteria than on gram-positive bacteria. Okay? So this is why people who have deficiencies in such proteins, okay, in the final common pathway of the complement activations, of the complement activation proteins, they basically are susceptible to infections of gram-negative bacteria way more than they are susceptible to, or more than other people. Okay? Now, there is actually, now why do, when we have a deficiency here, why can we kill gram-positive bacteria? Or how can we kill gram-positive bacteria without the use of the membrane attack complex? Now the thing is that if we just, C3b can act on certain receptors on macrophages or neutrophils. Let's suppose that this is a macrophage. Okay, now it has a certain receptor for the C3b. Okay, so if we have a deficiency from C5, etc., the C3b actually comes and binds with that C3b complex or C3b receptor on the macrophage or on the neutrophil, and it will enhance phagocytosis of that microbe or of that pathogen. Okay? So, important things to remember. We have two ways by which killing can occur: by the MAC or the membrane attack complex, or by just binding to the wall or to the receptor of the macrophage or the neutrophil in order to enhance phagocytosis. Now, phagocytosis does occur without this C3b, however, it takes a longer time, so it enhances phagocytosis. Okay? And so this is the classic pathway. Remember, classic pathway only involves the antibodies of IgM and IgG. I will see you in the next video.